The simplest organisms that we find on Earth are the bacteria group and the archaea group. These groups are both prokaryotes. They are the simplest cell type with no organelles, but their cells do contain genetic material and they can perform most of the basic functions that all of the organelles can perform in eukaryotes. We're going to start our discussion on bacteria by talking about some of their general genetic properties and also how they transfer genes from one to another. Then when we move into our next unit, we're going to talk about the diversity of these two groups. In general, bacteria are only going to have one type of chromosome. They're only going to have one copy of that chromosome. Most bacteria have a single circular chromosome, although there are some rare instances where some bacteria may have a couple of chromosomes. Usually they don't have very many, or they may have one or a small number of linear chromosomes. We're going to just assume that we're talking about a quote-unquote normal bacteria, and that bacteria is going to just have the single circular chromosome. We'll also talk about another type of chromosome called the plasmid, which is a small chromosome separate from the nuclear DNA. The chromosomes in a prokaryotic cell are going to be found in the nucleoid region. This is a region in the cell where the bacterial chromosome is packed in and generally stays, but it is not separated by a membrane like the nucleus in our eukaryotic cells. This image up here shows two nucleoid regions. This is most likely a cell that is getting ready to divide, and so it's already copied its chromosome. Down here on the bottom is an electron microscope image of the nucleoid region. Just a different image to show you where the nucleoid is found. Our prokaryotic chromosomes are double-stranded DNA, just like all of the other organisms on Earth. As I mentioned a minute ago, they are usually circular, and they're usually only a few million base pairs long, so they're relatively small genomes compared to eukaryotic cells that tend to have far more genes and a lot more chromosomes. They usually only contain a few thousand unique genes. Some have less than that, some have a little bit more depending on their size and their metabolic properties, but they're never going to have too many. And they're only going to have genes that are absolutely required. Remember that prokaryotic cells are incredibly tiny, and so they are not going to be able to physically maintain a large chromosome, and so they're not going to maintain genetic material that they don't need. They're going to keep those genes that they're using, and evolution will take out the ones that they are not. Most of these genes are going to encode proteins. If you've taken a genetics class or potentially a 101 class, you might know that not all of our genes encode proteins specifically. Some of them encode RNA, some of them are there for regulatory or structural purposes, but most of the genes in a bacteria are going to encode proteins, and there's going to be very little regulatory or structural sequences there. There is a single point of origin for replication. In a eukaryotic chromosome, there are multiple points where replication starts along the chromosome, and then that replication bubble opens up, and they sort of stretch into one another. In a bacterial cell, there's one single point. Replication happens from that point in both directions all the way around the chromosome until the entire chromosome is replicated, and then the two pieces, the parent chromosome and the daughter chromosome, are separated from one another and then partitioned into a new cell during binary fission, which is the mitosis version of division in a bacterial cell. I really love this image on the right. This is a bacterial cell that has burst and released its chromosomal DNA out into the environment. Some bacteria also contain a small piece of DNA called a plasmid. This is an independent piece of DNA separate from the nuclear DNA. They occur naturally in many types of bacteria. Some bacteria are more likely to have them than others, and they are found occasionally in some eukaryotic cells, but mostly they're going to be found in bacteria. Plasmids have genes that are not essential for survival, but they are genes that provide an advantage in particular growth conditions. So they might provide some sort of tolerance to a specific chemical or an ability to metabolize something special in the environment, just to sort of give them something a little extra to live in whatever conditions they find themselves in. Plasmids are also important because they can be easily transferred among bacteria. So because they're small and because they're readily selectable by natural selection, they can actually be transferred relatively easy. So for example, in a healthcare setting, we might have antibiotic resistance encoded on a plasmid that can then get copied by the cell, and then maybe when that cell ruptures, it releases the plasmid into the environment, and now other cells can pick up this relatively small piece of DNA with very advantageous genes on it. Plasmids have an origin of replication as well, and an origin of transfer. We'll talk about that in just a few minutes. And then they have a small number of genes that are going to be expressed by that plasmid. Plasmids may also sometimes be integrated into the genome, in which case they become a permanent part of that bacterial cell line. There are 5 types of plasmids. The first are the R plasmids, or the resistance plasmids. These are plasmids that are going to help the bacterial cells survive in situations where they might find themselves in the presence of antibiotics or other toxins. So basically they're going to allow the bacteria to produce a particular protein that is going to break down the antibiotic or toxin so that they can survive in that particular area. Bacteria may also have degradative plasmids. These are similar to the antibiotic or toxin plasmids, except that they are basically a metabolic use. They are digesting or using something unusual in the environment. So maybe they live in the environment where there's a particular chemical that's not very common somewhere else, or a particular mineral or something like that. Col plasmids encode colicins. These are proteins that are essentially antibiotics against other bacteria. So they are basically the bacteria producing an antibiotic that is going to kill other bacteria in that region. A virulence plasmid is a plasmid that contains genes to make a bacteria pathogenic. Pathogenic means that it can spread disease, or it carries a disease that can be spread. And virulence means that the bacteria has the ability to spread that disease. So sometimes that ability is conferred on a plasmid, and when that is the case, it can be transferred to other bacteria who then become pathogenic and can then themselves spread that virulence plasmid. Lastly, we might see a fertility plasmid. This is called an F factor, and we are going to see this in just a minute when we talk about our methods of horizontal gene transfer. A fertility plasmid is going to allow the bacteria to produce a special tube called a pilus, and that pilus is going to be used to transfer genetic material from one bacteria to another. Bacteria reproduce really quickly. They reproduce by a mechanism that is relatively similar to mitosis that you would have learned about in Biology 101, although it's not quite as complicated as mitosis, and it is not exactly the same thing. But the idea is the same. During the process of binary fission, The bacterial chromosome is going to be copied. Then the cell is going to be pinched in the center. The plasma membrane is going to pinch in, and then a cell wall is going to form between the two cells of the bacteria, and then they're going to become two. In binary fission, each cell is a clone of the parent. So unless there happens to be a mistake in the DNA replication, they should be exactly identical to the parent. And during bacterial reproduction, the plasmids are basically going to just get partitioned in the cytoplasm, wherever they happen to be. But remember that plasmids have their own origin of replication, and so they can replicate independently of that bacterial chromosome. So as long as any of the plasmids that are in the parent cell are partitioned into the daughter cells will have those as well. In the image on the right, over here we have a bacterial plate, and you can see on this plate that there are individual little spots of bacteria. Those are called colonies, and they are plated in such a way that the colonies are spread out so that they are isolated. That way they can be picked up for study or for further isolation, further growth, separately from any others. Each colony is going to indicate the presence of a single bacterium, and then each colony's characteristics are going to be different depending on the type of bacteria. This one looks like it's probably E. coli. E. coli is usually a kind of creamy yellow color. It makes these small yellowish cream colonies on nutrient agar.